Hello, and welcome to everyone joining us for today's webinar, Challenges of Drug Development in Early Parkinson's Disease. The presentation runs for approximately 40 minutes. Please feel free to submit questions and comments for our speaker throughout the presentation using the questions chat box. We're a leading global contract research organization that partners with biotechnology and pharmaceutical companies to create custom solutions that advance new medications from discovery to reality. Worldwide Clinical Trials is dedicated to advancing clinical research to improve patient outcomes with neurodegenerative disorders. Our full-service clinical development capabilities include bioanalytical laboratory services and Phase 1 through 4 clinical trials, as well as post-approval in real-world evidence studies, all powered by a talented and accessible team of more than 3,500 professionals in over 70 countries. Our speaker today is Dr. Tomislav "Tom" Babik, a board-certified neurologist with over 40 years of clinical research experience specializing in Parkinson's disease and other neurodegenerative disorders. Dr. Babik has been with Worldwide Clinical Trials for the past 2 decades, where he manages the scientific aspects of clinical trials in neurodegenerative diseases. He has a profound understanding of the challenges faced by patients and caregivers and the complexities of clinical trial design. His extensive expertise and global perspective make him an invaluable asset to the field of neuroscience research. We are thrilled to have Dr. Babik with us today to share his insights on the latest developments in early-stage Parkinson's disease research. Without further ado, please join me in welcoming Dr. Tom Babik.
Okay, good afternoon. It's my pleasure to present you the challenges of drug development in early Parkinson's disease, which is hot topics definitely for many years, because we know that treatment of Parkinson's disease was well established 25 years ago, but unfortunately in the recent few years there is no update which we should be happy with. I'm going to cover particularly challenges in patients with no treatment and, of course, patients who are participating in so-called disease-modifying clinical trials. Of course, there are plenty of unmet medical needs in treatment of Parkinson's disease, which are including particularly disease modification, neuroprotection, also alleviation of non-motor symptoms, which are common in Parkinson's disease. Also, in advanced stage of Parkinson's disease, we need to improve gait and disequilibrium problems. And of course, because many of these patients are taking either levodopa or dopaminergic agonists, there are plenty of long-term side effects of levodopa and dopaminergic agonists, which we are calling levodopa-induced fluctuations, and definitely there are plenty of space to improve treatment in these patients. Finally, quality of life and caregiver burden are very important aspects of the treatment of Parkinson's disease. At the next slide, I'll share with you the problem of early Parkinson. I can share with you the problem of clinical symptoms of Parkinson's disease. Now we can calculate, we can basically divide Parkinson's disease in 4 different stages, which are including prodromal Parkinson's disease, early Parkinson's disease, mid-stage of disease, and the late stage. Today, I will be focused mainly on early Parkinson's disease, which are including motor symptoms and also usually plenty of non-motor symptoms, characteristics for these patients. I will not talk about diagnostic procedure, but I will share with you some important aspects of prodromal Parkinson's disease as well. At the next slide, there is a schematic review of the prodromal Parkinson's disease. You can see that prodromal Parkinson's disease basically starts many years before diagnosis. So in fact, there is an estimation that about 75% of dopaminergic neurons should be destroyed when we have typical motor presentation of patients where we can make a diagnosis of Parkinson's disease. However, there are plenty of risk factors for developing of Parkinson's disease, which are including age, family history, environment, also smoking history, genetics. And during this period of prodromal Parkinson's disease, there are few, I would say, biological markers which we can consider as a potential heralds of Parkinson's disease, which are including anosmia, poor olfactory sense, constipation, REM sleep disorder, autonomic problems, and very often we have anhedonia or depression in this stage. However, diagnosis of Parkinson's disease should be made clinically with the occurrence of specific early motor symptoms, which are including bradykinesia as a mandatory sign and tremor and rigidity as a 1+ symptom. Of course, we want to start treatment as early as possible in patients with Parkinson's disease, and also we have today few drugs which we are calling symptomatic drugs for treatment of Parkinson's disease. However, we need to try treatment as early as possible, and what we need a disease-modifying approach, which we are commonly called neuroprotection in patients with Parkinson. However, there are plenty of challenges in clinical trial design for neuroprotection, and of course, this is a consequence of imprecise pathogenesis of Parkinson's disease that we are not aware of in many aspects. Also, there is no ideal animal model which could present Parkinson's disease and reflect slowly progressive neurodegeneration in humans. Also, Parkinson's disease has a various progression, and which is often age dependent. So early onset usually have a faster progression than late onset. Also, availability of various pretty effective symptomatic treatment at the beginning of treatment, of course, compromise design of the clinical trial, and we are not sure was it consequence of the clinical trial, a new product, or symptomatic treatment. Of course, we do not, we basically need to establish long-term safety profile of the investigation product, and of course we are always faced with levodopa complications in our patients, and often we need to treat this complication on the right way, which is also complicating a disease neuroprotection clinical trial, which is quite long, about 2 to 3 years. At the next slide, I'll show you two different aspects, two different approaches to the treatment of patients with Parkinson's, with early Parkinson's disease. One is a well-known concept called the honeymoon, which is basically standard well-established treatment of patients with diagnosis of Parkinson's disease. So practically, after diagnosis of Parkinson's disease, many of these patients will be prescribed levodopa or dopaminergic agonist, which is at the start of symptomatic treatment. And this honeymoon period is basically called honeymoon because usually majority of patients in the beginning of treatment have a very good response to the treatment, which will last a year, 2 or 3, but this effect should subside, and that's why we have so-called levodopa wearing off phenomenon, where levodopa effect is going to disappear, and we are faced with either complication or more dose of levodopa should be prescribed to keep the status before. On the other side, what is important for the clinical trials is so-called golden year concept introduced by Robert Hauser a few years ago, which basically describe a latent period after diagnosis, which is so-called symptomatic treatment-free period, where basically patients could participate in clinical trials with no levodopa or no dopaminergic agonist, where new drugs should not be compromised by very potent symptomatic treatment. Today, I will talk a little bit more about golden year. I'll talk what is the percentage of investigators and physicians happy with such a concept, and how long we can basically keep these patients without symptomatic treatment at the beginning. There are a few clinical trials before when we've seen that practically even after 9 months or a year delay of symptomatic treatment, we basically, after 4 to 5 years, have exactly the same patient status. So these clinical trials are basically justifying start of symptomatic treatment after golden year. We made, a few years ago, we made search and interview with more than 250 neurologists in more than 30 countries all over the world to understand what is the standard approach of these investigators and physicians in treatment of Parkinson's disease. So the question was, how often do you delay starting levodopa dopaminergic agonist in newly diagnosed patients with Parkinson's disease? So you can see the percentage of various patients group, which are including patients with the onset of disease before year of 60 and after year of 60. And you can see that practically 15 to 30% of investigators in both conditions are using delay of the levodopa treatment. They are using so-called symptomatic treatment-free period. Often we can find practically in the same percentage, whereas rarely we can see that practically 50% of investigators with the patients younger than 60 are basically using a levodopa delay process. Similarly, the question was, how often do you delay starting levodopa and dopaminergic agonist in de novo Parkinson's disease patients who are under some sort of treatment, which are usually MAO-B inhibitors, amantadine or similar. On the right-hand side, you can see the percentage of answers as well. And the next slide, there is a review of the most common initial treatment of newly diagnosed patients in Parkinson's disease. Practically 70% of all of investigators interviewed using MAO-B inhibitors. We have evidences that MAO-B inhibitors might be used as neuroprotective agents in Parkinson's disease, although the effect of MAO-B inhibitors is quite weak, whereas amantadine was used in about 17% of patients with early Parkinson's disease. Amantadine was pretty old drug and also widely used in advanced stage of Parkinson's disease for the treatment of dyskinesia, but it seems because of its cholinergic action and in some respect neuroprotective agents, quite a large group of investigators are still using amantadine as a first treatment of Parkinson's disease. Anticholinergics and others are pretty rare, where 7% of investigators are not using any of these compounds before starting treatment of levodopa or dopaminergic agonists. What is the main reason for initialization of levodopa or dopaminergic agonists in our patients? It's quite interesting review that practically 77% of investigators are starting levodopa treatment in early Parkinson's disease upon patient's request, which is very interesting, you know. So objective data, which are coming either from UPDRS3 or 2 deterioration or patient's quality of life, are less important for the treatment, for the initialization of symptomatic treatment. We also investigated the experience of our investigators in Parkinson's disease clinical trials, and we asked about experience in early Parkinson's disease. And you can see that 92% of our investigators have experience in phase 2 and phase 3 clinical trials in all types of Parkinson's disease. However, in de novo Parkinson's disease with no treatment, 74% are experienced, whereas in early Parkinson's disease, de novo treatment with one or more drugs is less common in terms of patient of our investigators' experience, but this is pretty good review that practically de novo Parkinson's disease with no treatment should be seen in the record of the investigators in about 75% of all of them interviewed. Also, we asked what is the percentage of neurologists who will delay symptomatic treatment if needed for a clinical trial. And up to 3 months, there are 27%, up to 6 months, about 50%, and up to 9 months or a year, in between 4 and 12%. 8% of patients, 8% of investigators would not delay treatment at all. So practically, these investigators are not suitable for clinical trials with early Parkinson's disease with no levodopa or dopaminergic agonist treatment. It's interesting that expected average delay of symptomatic treatment among investigators, our survey showed about 5.4 months. So practically nearly 6 months, we can easily design no symptomatic treatment period clinical trial in newly diagnosed patients with the clinical trial with the new disease-modifying treatment. We also investigated what is the main challenges for the recruitment of patients with de novo Parkinson's disease in randomized clinical trials. So we found, we had to use analog scale between 0, no challenge, and 5, the biggest challenge, and we've seen that complexity of study protocol probably has the minor effect on the recruitment, whereas the highest negative effect of the recruitment are patients' willingness to participate in the study. You know, patients are taking a lot of information from various sides, from web, from families, from other patients, and they have their own opinion about rationale of clinical trials. Therefore, having in mind what we've showed so far, the investigators selected for the clinical studies are the most important people who will talk to patients and explain to them pro and con aspect of the participation in early diagnosed clinical trials in Parkinson's disease. There are several different approaches to disease-modifying design studies in early Parkinson's disease, well described by Tony Lang 10 years ago, and nothing changed so far in the last 10 years. So there are a few different types, which are including classical parallel design or washout design or delayed start design. All of them have a pro and con aspects. But recently, we basically are faced with many of studies which are using a classical parallel group study design with futility in double-blind placebo-controlled clinical trial with usually 18 to 24 months drug exposures to the patients and placebo as well. In many of these studies, symptomatic treatment should be allowed after certain months of certain months of participation, usually after 9 or 12 months, and also having in mind that, based on the previous clinical trials, that in the long-term prognosis, after 3 to 4 years of treatment, these patients would be practically at the same level as those who had a symptomatic treatment from the beginning, from the diagnosis. In early patients and disease-modifying treatment, the selection of endpoints are very important. And of course, we are using in symptomatic treatment, we are using UPDRS or MDS UPDRS recently for the assessment of the drug effect. FDA is favorizing UPDRS Part 2, which is activities of daily living of patients with Parkinson's disease, as a main endpoint in patients with disease-modifying treatment. And particularly slope analysis. Recently, they also introduced the concept of the time to the important event as a primary endpoint, usually using ADL, which is UPDRS2, or combining UPDRS2 and UPDRS3. This is important in planning the clinical trials, and particularly sample size, that we need to understand what is the minimally clinically important difference in patients with Parkinson's disease after a certain amount of months exposure. At the next slide, I'll show you what are important recruitment components of disease-modifying clinical trials in de novo patients. So basically, it's very important to have excellent infrastructure, which are including a research team, which basically are using recruitment plan, recording the recruitment activities, communication with the patients, and also recruitment toolkit. Protocol complexity is also one of the important aspects of the recruitment, which protocol should be simplified with a clear study design, entry criteria, and outcome measure. Concept, which investigators should be used? Of course, those who are preferring a golden year approach to the patient treatment, and also as investigators who are experienced in the clinical trial with de novo Parkinson's disease. And finally, patient's characteristics as a very important. Those who are accepting the golden year concept versus honeymoon treatment, patient's altruism, and also patient's trust and personal health benefit. And the next slide, important for the recruitment and for the planning. Many of sponsors are too optimistic in terms of the recruitment rate in early Parkinson's disease. We can see here on the left-hand side what was the recruitment in published clinical trials in early Parkinson's disease, which are between 0.2 to 0.6, depends on the allowance of dopaminergic agonists or not. What is even more important is source of patients. So one study designed particularly to review source of patients who participated in clinical trials concluded that practically 80% of patients recruited in a study are coming from the site personnel. So basically, talking around from the site personnel is the main source of the patients in a clinical trial. Neurologists and uh, trial finders are less important, whereas others, which are website information, clinical trial government.com, patients group website, are definitely less important. So neurologists and site personnel are definitely the most important source of newly diagnosed patients who are participating in a study. A few words which are important in terms of the design of the clinical trial, which are including minimal important differences in early Parkinson's disease. This is, in fact, important to use either MDS UPDRS or classical UPDRS. The difference is that MDS UPDRS is slightly extended, although there is a possibility to validate and to compare these two assessments using specific mass form. But it's important to say that clinically important difference using ADL, for instance, should be 2.5 points in 1 year. I had the opportunity to talk to one of the sponsors a few years ago, who basically set up main endpoint time to difference of 1 point. 1 point is usually 1 point difference in ADL is usually a natural curve of progression. So practically majority of patients will deteriorate after 1 year on the treatment, 1 point at ADL. But 2.5 is clinically meaningful. So therefore, they need to plan study at least 3 years to see the difference, which will be clinically significant. However, statistical significance can be obtained with less points difference. Recently, biomarkers are becoming very important as longitudinal measures in patients with Parkinson's disease. We have several biomarkers which we are using as a diagnostic biomarkers. Particularly, this is DataSpect, which is important help for a diagnosis of Parkinson's disease. But also, there are several biomarkers which will help us to make distinction between Parkinson's disease and Parkinson plus syndromes. And of course, these biomarkers can be obtained either from the imaging or from the biochemical assessment of the different on the different fluids. At the moment, there is no biomarker in Parkinson's disease which should serve as a longitudinal biomarker, which will be accepted by FDA as a surrogate biomarkers, as we've seen Alzheimer's disease with beta amyloid. To conclude this review, we can say that patients' willingness to participate in clinical trials and delay of symptomatic treatment are the most important challenges for the successful recruitment in randomized controlled clinical trials among de novo Parkinson's disease patients. We can also confirm that delay of symptomatic treatment in de novo Parkinson's disease up to 6 months for clinical trial purposes seems acceptable by 2/3 of investigators. And of course, initialization of symptomatic treatment in de novo Parkinson's disease in clinical trials is mainly driven by patients' wish rather than objective findings. So therefore, there is definitely ethically, there is from the ethical perspective, delay of the starting treatment, which we are calling symptomatic treatment free, can be accepted in design of the drug development in early Parkinson's disease. I would stop here.
We have reached the end of our presentation. If you have any questions, please don't hesitate to contact Tom using the contact information on the screen. We sincerely appreciate your time and interest in our webinar. Thank you for attending.